Well, hello once again, students. So at this point, I'm hoping you have kind of an understanding of how glucose is broken down into carbon dioxide. We can see that it's much more complicated than just a single reaction. There's a series of 10 chemical reactions in glycolysis where glucose is broken down to 2 molecules of pyruvate. There's this pyruvate oxidation transition reaction where that pyruvate is converted into a 2-carbon acetyl coenzyme A. And then there's this Krebs cycle where that acetyl coenzyme A is further broken down to CO2. So the gist of what we've been talking about so far is that ultimately that glucose has been broken completely down into 6 molecules of CO2. It's completely ripped apart. It's destroyed. Why would we destroy this lovely molecule? Well, in destroying that molecule, we are taking those carbon-hydrogen bonds and forming carbon-oxygen bonds, which releases chemical energy. And we are coupling the breakdown of that glucose to CO2, ultimately with the production of ATP. So we have harvested the chemical energy of glucose into the chemical energy of ATP. Along the way, we've also captured some of that energy as these reduced electron carriers, NADH and FADH2. So our final phase here is we need to deal with these electron carriers. NADH is not the energy currency of the cell. ATP is. So we want to get rid of that NADH and use it to make even more ATP. And this column we've been ignoring for a while is foreshadowing because we can see that ultimately this 2-NADH is going to be used to give us some ATP. And same with all of this. So the question is how? Well, for that, we need to talk about the electron transport chain. This is a series of proteins that pass electrons from NADH and FADH2 to oxygen, and that uses the energy released to make ATP. So essentially, we're passing an electron from NADH and FADH2 to oxygen, and then harvesting the energy released to make ATP. The electron transport chain is located in the inner mitochondrial membrane. You may recall that there's 2 membranes to the mitochondria. It's in the inner one. And we can see that here. So here's our mitochondria. Here is that inner membrane. And lo and behold, we see these proteins that make up the electron transport chain right in that inner mitochondrial membrane. And then we also see, as I said, that these NADHs, we're finally going to figure out how those are used to make ATP, and the FADH2s are used to make ATP. So we're going to do players process here because, again, it gets very complicated. So the first players are these electron carriers, right? NADH and FADH2. We saw that these are produced during glycolysis, during the Krebs cycle, during the pyruvate oxidation transition reaction. They're produced all over the place. We can get the NAD plus, like the starting ingredients, we can get from vitamins. NADH, remember we said, was from niacin. FADH2 is from riboflavin. So those play a role. Second are these electron transport proteins. And by the way, this picture is in your packet, and so you should have this diagram. So the electron transport proteins. Now, if you look at these, review from previous units, are these integral or peripheral membrane proteins? Integral? Did you say integral? I hope you said integral. They're integral. So they are passing through the membrane here. And then what are those foldings of the inner membrane called? So we said that the mitochondria has these little infoldings. And do you remember the name? They're called cristae. Cristae. And their purpose is to increase the surface area. And that should make sense because if these proteins are involved in making energy, gosh, we'd want a lot of room to put a lot of those proteins. And that's why we have this folded membrane. What I want you to think about, though, is as these proteins are kind of just drawn in a row here, here's just another diagram that kind of shows those proteins on almost like a graph where it converts the free energy relative to oxygen. And you can see that NADH has the highest, but then each protein is slightly lower, and then oxygen has the lowest. And so essentially, as electrons are passed from NADH through these series of proteins to oxygen, energy is given off. And again, this hill analogy is a useful one. You can just kind of think of each of these proteins, as this little intermediate carrier of that energy. So instead of just like shoving that electron all the way down the hill, it's kind of passed through this series of proteins slowly and in controlled. So essentially, the potential energy of the electron is decreasing as we go from NADH through these proteins and ultimately to oxygen. And that free energy that's released is what is ultimately harvested to make ATP. And then one other little point, remembering what you know of membrane structure, do you think these proteins are arranged in a specific order within the membrane? In other words, are they arranged like this where there's kind of rigidly locked in place? This first one here, second one here, third one here. And hopefully you know the answer is no, because remember, the membrane is constantly moving around. It's a fluid mosaic. And so in reality, these proteins are all wiggling around all over the place in the membrane. And in order for them to pass the electron, they have to bump into each other. And so this is the order in which they need to bump into each other to make things happen. It's not like they're rigidly locked in place there. They're all wiggling around and ultimately will bump into each other. So just kind of keep that in mind too. The next player are the proton pumps. So some of these electron transport proteins are also proton pumps. Now you may think, wait, proton, H plus, is that a proton? But if you think about a hydrogen atom, it has one proton, one electron. So if it's H plus, it's lost its electron, which means it's just a proton. And so the proton pumps are these electron transport proteins that couple the exergonic transfer of electron from one protein to the next, again, down the hill. They couple that exergonic electron transfer to the endergonic transport of a proton across the membrane. Now, why would it be endergonic? Well, the inner membrane space here, this space between the membranes, actually has a high concentration of H+. And so these proton pumps, they are moving H pluses against their gradient into this inner membrane space. That's endergonic, and so it's coupled with the exergonic movement of that electron. Okay, so some of these electron transport proteins are also proton pumps. Another player is oxygen. So oxygen is the final electron acceptor at the end of the electron transport chain. And again, we see oxygen here. It is the final electron acceptor. And this is why, by the way, you need oxygen. So every time you inhale, that oxygen you're taking in is being used for this process right here. That's it. So really the only reason you have lungs is so you can take in this oxygen that's needed here and get rid of the CO2 that we've already discussed. Another player, ATP synthase. So ATP synthase is a protein that couples the exergonic movement of H plus down its gradient to the endergonic production of ATP. So this protein right here, it's going to make ATP, but that takes energy. And the energy used is the endergonic movement of H plus back down its gradient. So earlier we had the endergonic, requiring energy, movement of H plus against its gradient. Here we have the exergonic movement of H plus down its gradient. So complicated. Here's just another view of this same thing. Here's our electron transport proteins. Here's the NADH donating electrons. The arrow indicates the movement of electron, ultimately to oxygen. And then you can see that some of these are proton pumps moving the H pluses against their gradient. And then here's ATP synthase where the H pluses are moving down its gradient. So again, we're still in the players. This should still be kind of foggy. So let's take a look now at the process of how this all goes down. So the fancy word for the process here that we're about to discuss is called chemiosmosis. So here's a whole bunch of text that sort of walks through the process. And I think that text is useful. And so you may want to pause and make sure you get all that written down. But let's kind of break down what's actually happening. So the first step, NADH and FADH2, again, these were from the Krebs cycle, from glycolysis, from pyruvate oxidation. These donate electrons to specific electron transport proteins. And so we see that happening here. NADH is going to donate an electron to a specific protein. And when it does that, NADH becomes NAD+, and that NAD+ is going to go back to the to glycolysis, Krebs cycle, to be reformed into NADH again. So that's kind of recycled later. So NADH donates its electrons to a specific protein. FADH2 does the same thing. It donates electrons to a specific protein. Those electrons are passed down a chain of proteins, each slightly more electronegative than the last. So these electrons, once they're passed on, is going to be passed from protein to protein to protein. At the proton pumps, the energy that is given off by the movement of that electron is going to be used to transport protons against their gradient. So as the electron moves from protein to protein, energy is given off. That energy is used to pump protons against their gradient. Okay? Oxygen is then going to be the final acceptor of those electrons, and when it accepts the electrons, it actually forms water. So the electron goes from protein to protein to protein, finally being accepted here by oxygen, and then oxygen joins with two protons as well to form water. Okay, and try not to get too hung up on the chemistry. If you're a chemist, you can, your textbook, I think, might have better pictures to kind of really digest the chemistry. But essentially, the oxygen is undergoing a chemical reaction where it gains an electron and some H pluses and forms some water. Okay, those H pluses now, we've kind of piled them up in that intermembrane space. They're going to move back down their concentration gradient by passing through ATP synthase, and that energy released is going to be used to make ATP. So here's the proton. It's going to now move back down its gradient, and that energy is going to be used to make ATP. So again, NADH donates an electron. That electron gives off energy as it's moved from protein to protein to protein, ultimately reacting with oxygen to form water. Along the way, the energy release is used to pump protons against their gradient. Those protons then go back down their gradient, and the energy released as they go back down their gradient is used to produce ATP. That's the gist, okay? Some accounting. Each NADH pumps about 10 protons across the membrane. Each ATP requires about 4 protons to move through ATP synthase, and so if you do the math, that means that there's about 2.5 ATP per NADH. And so we see 2 NADH times 2.5 gives us 5 ATP. FADH2 gives us a little bit less energy, so those 2 FADH2s end up giving us 3 ATP. And so again, you can see all of that in this overview diagram. Here's that movement of electron. Here's the protons being pumped into the intramembrane space. Here's those H pluses going through ATP synthase to make the ATP. And then here's just kind of the mathematical accounting of how much we get. So that's pretty much it for the electron transport chain. Complicated. We're going to be working with it a little bit more in class, give you some hands-on activities, hopefully, to help you work with it. But the efficiency of cellular respiration overall is about 34%, which is pretty darn good. If you remember, glycolysis, I think, was about 1.5%. Now, if we do all of these steps, we're up to 34%. So we get about 34% efficiency when we convert glucose into ATP, which is pretty darn good. So I'm going to pause there. We got one more screencast to tie up some loose ends and look at some special topics around this. So we'll continue it there.